This is a production of Cornell University.
Well, awesome. Thanks so much. It's a pleasure to be here. While I was preparing for it, I was realizing that it's almost been 10 years since I first took soil science with Jonathan Ruslanelli. So it's been quite a while being connected with this department and learning from all the faculty in this department. So it's a pleasure to be giving this seminar, and of course weird to be giving it virtually and not being able to see your reactions and all that. But I hope something funny comes up in the transcript as I'm talking, because I already saw some funny things. So with that, I'm going to be talking about characterization of soil health in New York State, the effects of soil texture and inherent property and cropping system, the effects of management, and also some soil health data analytics work that we've been doing in the lab nationally to improve soil health assessment. So the presentation is mostly going to focus on this characterization of soil health in New York State, which is just published in these summary and technical reports. Some of the more recent work that kind of connects off of that, which is some more regional soil health analyses in Western New York and Long Island, um, partnering with collaborators in those areas. And then the data analytics part is going to focus mostly on using random forest to model available water capacity and ACE protein to help us simplify soil health testing and make it cheaper. So here, just to, if you haven't looked at the report already, here's the characterization of soil health in New York State. There's a summary report, which is shorter, and technical report, which is long and has umpteen tables in it with a lot of data. So definitely check those out. Those have both been submitted as peer-reviewed manuscripts, so hopefully we'll be seeing those sometime in the future. And obviously we need to acknowledge New York State for funding this and everyone that contributed to this. But for those that maybe don't have a soils background, we know that soils have inherent and dynamic soil qualities, and these inherent soil qualities is what Jonathan and soil pedologists spend their time studying. It's the combination of these parent materials, climate, biology, relief, and time in forming soils. And this is one of my favorite soil pictures of soil profile, Hagerstown soil profile, and it's extremely high res, so good for teaching in a virtual setting. And then we have these dynamic soil qualities, which are changes due to human use and management. And when we talk about soil health, we're really talking about these dynamic and anthropogenic aspects of soil quality. So you've seen Harold present a couple of these slides, so I'm going to go through this, but we all recognize the importance of soil health through its ability to function and provide services. And we can really think about the importance of soil health through two lenses, both through kind of the agriculture and plant and animal health, nutrition side of things, and the ecosystem services side of things, water quality, climate regulation, and on and on. But more and more, what we're starting to think about in the Anthropocene is humans' tremendous influence on soil resources. So yeah, this interest in soil health is driven by this renewed recognition that soil is critical for climate, for biosphere integrity, for water quality, for food security, and we need to understand these increasing anthropogenic impacts on a planet's not getting any bigger. So I don't need to go into too much detail on this. You guys are familiar with the Soil Health Lab. It's been around for a while. Through the years, the current iteration of the port is really a nice thing to look at, and focuses on some practical indicators that the lab decided to go with over the years of researching many indicators, and interprets measured values within different soil texture groups, and provides guidance for management and constraints. And we've measured, the lab has measured a really large number of samples. So what we've been up to recently is really trying to get at um, um, using that information both nationally and in the state to improve soil health testing and interpretation and benchmarking and all those things. So what we're going to spend the most of the time on is this recent characterization of soil health in New York State report. We had 1,456 composite soil samples from 0 to 6 inches from across the state. You can see the distribution of those samples. And this is not a statistically designed study, but kind of using modern data analytics to look at these trends. And the indicators we're going to talk about today, we did the characterization of all the soil chemical properties as well by soil texture and cropping system, but today we're going to focus on the physical, which includes available water capacity, aggregate stability, surface and subsurface hardness, which is the only one that's measured in the field with a penetrometer. Important to have the right conditions for that measurement. And then the biological, which is total soil organic matter, active carbon, which is kind of the fraction of easily oxidizable organic matter, soil protein, which is the most important nitrogen containing compound in the soil, so an indicator for the organic nitrogen pool, and soil respiration, which is a measure of metabolic activity. So the first question, which we've known a lot about as soil scientists, but is always good to get a handle on for our region. So we wanted to understand how soil texture, which is probably the most dominant inherent soil property, influences these soil health indicators. Because if the point of soil health assessment is to be able to assess management effects on soils, we really need to understand these baselines, so we're not comparing apples to oranges. So you can kind of see we have maybe dominant, a lot of loams in our most productive soils, more sandy soils in Long Island, but I won't go into that so much. And we know some of these indicators are really influenced by soil texture, and we recreated this graph from our data, and this could easily go into Brady and Weil textbook. It's exactly what you'd see in there, that the medium textured soils, especially the very silty soils, tend to have very high available water capacity. And we've also known for a long time that soil texture controls the amount of soil organic matter that a soil can hold. Basically, fine silt and clay particles help stabilize organic matter against decomposition. So the more of those fine particles you have, the more organic matter you can stabilize against decomposition. So when we look at this huge range of soils from across New York State, you can see pretty sizable differences between coarse loam, silt loam, and fine texture groups. And this information is really important because we need to be able to interpret within the texture group. Otherwise, we're looking at differences that are going to totally mask the effects of management. And additionally to organic matter, I think what's a more novel contribution is describing these more novel biological indicators by soil texture as well. So we have active carbon and respiration, which also follow those same trends. And so I think the biggest contribution of this report and this research is to really try to get at how do cropping systems, farmers management impact soil health. So what we did here is when every farmer submits a soil sample to the soil health lab, they put in their crop codes of the previous 2 years and this year. And what we did was basically categorize those into 5 different cropping system groups. So the annual grain brought together corn, grain, soy, wheat. Dairy brought together corn silage and alfalfa crop codes. Pasture, different pasture crop codes. Mixed vegetable is a mixed crop code. And the processed vegetables is a single vegetable crop code. A lot of snap bean production, summer squash, transplanted cabbage, potatoes, pumpkins. And this distinction was really important that we wanted to make because we wanted to distinguish between the effect of management in these smaller, maybe less than 5 acre mixed vegetable farms versus the much larger scale processing vegetable farms, because you can probably imagine it's much easier to influence soil health in a couple acres than it is in 100 acres. So that's what we did, and you can see kind of the distribution, the proportion by county of these different cropping systems across the state. Um, Kind of where our soils are the best, from Buffalo to Albany. We have where most of our annual grain production is, and most of our processing vegetables, and also a lot of dairy in Western New York. Northern New York, mostly dairy, um, and Hudson Valley and Long Island has more maybe mixed um, vegetable production. So we're a very diverse state in terms of different types of systems. So it's not as easy as it would be in the Midwest to try to characterize, but also helps us find some differences more easily. So we saw this before, so I won't go into it, but the significance of this is basically that these different cropping systems have extremely different carbon and nutrient cycling regimes and tillage regimes that are basically influencing this. In pastures, we have 90 to 100% of the biomass being returned to the soil. We have no disturbance through tillage. So all so that organic matter is able to persist in the soil a little bit longer, and we're not disturbing soil microbial communities. On the flip side, in cash crops, processing vegetables, and annual grain systems, we are exporting a huge amount of the carbon and nutrients from this system. A lot of these systems, we still have a lot of tillage in New York and a lot of erosion, also taking um, topsoil, and organic carbon from the system. And kind of intermediate to that, figure that these dairy cropping systems that have these longer rotations of 3 years of corn silage, 3 years of alfalfa, um, are applying a lot of manure, have perennial forages, or maybe having a net input of carbon and nutrients because they're importing feed onto the farm, and then mixed vegetable farms, which um, usually smaller in size and some of them, a lot tend to be organic, so they're using compost for their fertility, and a lot of cover cropping. So here we're going to look at the effect of these different cropping systems on different indicators. And right now you can see that we're just looking in the loam texture because we want to be comparing apples to apples. We know that soil texture has a huge effect on that indicator and those other biological indicators. So here we see that, pastures and mixed vegetables are able to maintain a much higher level of soil organic matter than annual grain and processing vegetable systems, and dairy crops were somewhat intermediate. Active carbon by cropping system, so pastures, dairy crops, and mixed vegetables tended to have higher amounts of easily decomposable organic matter for microbes compared to the processing vegetable. And what was interesting is these are both the same samples, but dairy crop tended to have lower organic matter than mixed veg and processing veg in pasture, but similar amounts of active carbon. So I don't exactly know what that means, but interesting nonetheless. Soil protein, similar as total organic matter, much higher in the pasture and mixed veg systems than dairy crop, annual grain and processing vegetables. And this is an indicator of that organic pool of nitrogen in the soil. Soil respiration, the pasture soils had a very, very high signature for soil respiration compared to these other indicators. And we could talk about that, why that is if people are interested in that. And what's really nice, proof of concept, I think a lot of people tend to just think about the biological when we talk about soil health, but aggregate stability is a really great indicator for soil health, and is very sensitive to these different cropping systems. So a couple things is going on here. We first know that, yeah, undisturbed systems with no tillage that have living roots all year, their mycorrhizal fungi are intact, and those fine roots and mycorrhizal fungi are just really huge actors in building and maintaining aggregates in the soil. And we also know there's a positive relation between organic matter and aggregate stability. So that could explain why these mixed vegetable and dairy, which could could conceivably have the same amount of tillage as the other systems, but are maintaining a little bit higher organic matter. And this is, yeah, nice proof of concept because big data set showing this in all texture groups. You could probably pretty reliably go take a sample from a pasture, take a sample from a field that's been cultivated, maybe with low organic matter, and be able to do this slaking demonstration for your friend or a farmer to be able to show this concept. So when we look at all the soil health indicators by cropping system, we see they're all kind of telling us that processing vegetable and grain crops tend to have the lowest of these soil health indicators across the biological and physical. Dairy cropping are somewhat intermediate, and pastures tend to have the highest. So the next thing we did, which was interesting and also a novel contribution, was to do this variance components analysis, which is basically we have these two factor levels in our data set, this texture group and cropping system, and we want to understand which one is explaining the variance of these different indicators more. So basically what we saw is that soil organic matter and active carbon, the variance was about equally explained by both the texture group and the cropping system. Whereas for protein respiration, aggregate stability, the cropping system factor level was explaining much more of the variance in those indicators. Whereas texture, we saw earlier that very nice clean bar chart of the effect of soil texture on available water capacity. And it's a very texture controlled variable, which we'll come back to that, which enabled us to model it later. So what use is all this information? I guess before I did this, we could, having this information in the reports, we also have these tables by the different texture groups, and this can help us have a kind of a further level of interpretation than what we're doing now to be able to kind of relate your field. If you're a processing vegetable operation on a silt loam soil, compare that to the mean of this database and have some metrics for how you're doing compared to your peers in New York State. And we can also do a similar thing. So unlike air and water, where there's a lot of quality standards, those are more homogeneous medias, which are easier to get a measure. We have a difficult job as soil scientists of dealing with all the variability in the natural environment, but these metrics are really common and goals are common for other natural resources. So I think as we're realizing the importance of soil, we can use some of those, use some metrics and goals or whatever we want to call them to help farmers calibrate their management. And maybe these can be used in policy to help us target different goals. And we need it's really important for us to be at the table as soil scientists to make sure that these goals are kind of adapted to region, adapted to soil texture, adapted to cropping systems, because there's a lot of factors that go into it. We can't just say let's put a 2% soil carbon standard on all soils. So we can also yet develop these aspirational soil health goals by cropping system. And similarly using this database to basically target the 75th percentile. So 25% of the farmers are reaching this goal for this soil type and cropping system. So that might be a good goal to aim for if you were gonna aim for a goal. And we have these for these 4 different soil texture groups and all these cropping systems. So as we are feeling the impacts of anthropogenic climate change more and more, um, the public and a lot of people in New York State is really interested in building soil organic carbon as one climate mitigation strategy, um, to to address climate change. And of course, we know that soil carbon is the kind of keystone soil health indicator. So these things go hand in hand. We can do climate change mitigation and build healthy, productive soils at the same time. And kind of what we discussed before, soils greatly vary in their ability to store carbon, both based on the influence of soil texture, but also cropping system. You saw that cropping system seems to play a large role in how much soil organic matters in the soil. So we can use this information to help maybe target soils of greatest interest, of greatest benefit. So this is going to be a little bit detailed if you're not as close to this field, but I'll try to go slowly. So there's a bit of literature on carbon saturation and carbon saturation theory, um, and some equations to kind of predict the capacity based on other things. So this is just one approach. It's not perfect. The carbon saturation is definitely not is a little, the targets are a little bit elusive, but we can try to put numbers on it and show some things. So both these equations basically calculate the carbon saturation potential of the fine fraction. So the one that's more relevant for this paper is less than 50 micrometers. And basically we have the silt and clay content and use a regression equation to calculate that. And this is in this 6 et al. Paper. And these were basically data sets of grassland soils. So we're saying that these systems are probably at saturation, and there are linear relationships in those data sets with the amount of silt and clay content. So we can use those. There's a really nice paper that came out recently, which basically has a similar approach that we took for our New York data set, but in France, where they have huge amount of data, and did a similar kind of approach across different land uses. And then we could define the soil carbon deficit as basically this saturation potential minus what we reasonably have in that fraction. And I'm not going to go into this in detail, but you can see that the SOC sat minus SOC fine is going to give us our SOC deficit. And the most important thing is basically to show that the different cropping systems are further from this saturation point, which is this red line here. So these mixed vegetables and pastures are closer to that line, and some fields are very close to that line. And know there's a lot of the confidence. If I put the confidence intervals on here, it'd be quite large, but still a useful tool to show that annual grain processing vegetables, a lot of those fields tend to be much further from the potential. So there's much greater potential in a lot of these annual grain processing vegetable fields for building and sequestering soil organic carbon. So this is kind of an infographic we made for the report showing that. Additionally, yeah, Harold's already showed this, but if you're more graphic learner, this is basically the same concept, is that there's only so much capacity for soils to store carbon based on the concentration of silt and clay and the management systems. So we tend to know what works to promote soil health, conservation tillage, cover crops, perennial crops, organic amendments. So I think it's really about continuing to do the work we do in extension to promoting these reduced tillage and cover cropping practices um, to improve annual grain and processing vegetable systems and bring them up to what those other systems, which already have a lot of these components in them, um, to improve soil health. So perennial sods, manure, compost application. On the far right, I mean, what's really exciting in being in this extension field is the amount of thought and farmer innovation, research going into the integration of reduced tillage and cover crops. And we have our own expert here, Matt Ryan, who does a lot with no-till organic soybeans. There's a lot of interest in planting green, bio strip till, which is basically using precision ag to plant different strips of cover crops. One will winter kill, one won't, and you can plant your cash crop directly into that. So to wrap up this characterization of soil health in New York State, the key summary points are basically um, we can show the effects of inherent soil properties like soil texture on soil health indicators, which is really important baseline of information to have. And also that cropping systems with their associated carbon and nutrient cycling regimes and soil disturbance have a huge impact on these soil health indicators. And these metrics can help us um, benchmark farmers' soil health in a more kind of realistic cohort. So it's not as useful to compare a annual grain field to a mixed vegetable field, but being able to compare to the cohort. We can provide aspirational goals, and because we had such a soil texture rich database, we can kind of do some assessment of soil organic carbon storage potential across these different soil types and cropping systems. So next up, and this is work kind of bridging from this larger characterization of soil health. And this is work that's ongoing. I'm working with Debbie Aller in Long Island, Suffolk County, and Orleans Soil Water District, Soil Health, Western Soil Health Alliance, to basically probe into certain regions of the state. And with that, we can partner with people that know the operations really well, maybe help us improve and make our categorizations more local, and maybe they can help us with their relationships with the farmers get additional information, cropping history information, organic amendment input information to help fill out information and understand the effects of these management practices on soil health. So we'll start with Long Island. And there are no annual grain farms or dairies in Suffolk County, but Long Island agriculture brings different things. So there were actually some kind of woody plant operations. These are like Christmas tree farms and different things like that, and a bunch of perennial fruit. So those were also included as well. And this part of the presentation is definitely not refined yet. I might further refine these cropping system categories with Debbie Aller's input. But we can see similar things. Now we have the perennial fruit, but the single vegetable crop fields tend to be much lower than these mixed vegetable, perennial fruit, and pasture. And we're just looking at silt loam soils here. So here's just active carbon by cropping system in these pastures and perennial fruit operations, and mixed vegetables are maintaining higher amounts of easily oxidizable organic matter in the top 6 inches due to organic amendments or a lack of tillage compared to those processing vegetable systems. Respiration, a similar high signature for pastures compared to vegetable operations. Aggregate stability, this is just really interesting and nice to see how consistent aggregate stability is performing across these cropping systems. And yeah, obviously these undisturbed systems tend to have much higher aggregate stability than systems that are maybe tilled or have less organic matter. So now we're moving on to the Western New York part. And we've been, I should have only showed one side yet, but we've been collaborating with Franklin Egan and Sarah Nawa at PASA, and they've been doing this really large, maybe the largest of like a cohort, well, not the largest, but a large cohort in Pennsylvania and Northern Maryland of growers who are submitting samples maybe every other year, and then having them in different cohorts. And what they did is they did a bunch of individual reporting, which I really liked. So in addition to kind of the information we gathered, they were gathering things like days of living cover, how much are your organic inputs, and they were kind of plotting a farmer's field. So they would give this individual report back to the farmer and show how their organic matter inputs compared to their peers, or how their aggregate stability compared to their peers. And that relates to our Western New York project, because we have a nice collaboration with Orleans Soil Water District. In 2015, they submitted 45 fields, and then last year we sampled and resubmitted those fields again. So I did kind of a similar thing for a couple of the indicators, and kind of show where they were at in 2015 and 2020 and compared to their peers, because I think that's the most useful information we can give, because this, although it's anonymous, it's more personalized. These are the neighboring farms in Orleans County. So how do cropping systems impact soil health in Orleans County and some of the surrounding counties? You can see that most of the operations are annual grain or dairy or processing vegetable. So I think there's a lot of potential to refine these cropping system categories to make them more specific to a region. And we know there's much more complexity than just calling something an annual grain farm, a dairy farm, or a processing vegetable farm. There's annual grain, there's just processing vegetable farms, and there's farms that grow both annual grains and processing vegetables in the same rotation. There's people that just grow the cash grains. There's people that grow annual grains in a dairy rotation. There's people that are more strict dairy. So we're still refining this, but this is kind of the level that we want to go to be able to break out some of these trends. And this is maybe more useful for Orleans County, but also nice to see this proof of concept, the strong influence of soil texture on these biological soil health indicators. And what we're starting to see in this Western New York data set, this is for loam soils in Western New York, in Orleans County and for surrounding counties. Basically the systems that have vegetables in the rotation, things like cabbage and pumpkins, tend to have lower biological health. In this one, it showed up in active carbon and respiration compared to systems that are more dairy rotations, that might have a perennial forage or have the corn silage and potentially also have manure. So that was another piece of information that Katie Sommerfeld helped us extract, is whether these fields have history, a manure history. And you can see, despite these uneven sample sizes, that the fields that had received manure had higher soil organic matter and respiration than if they had not. Okay, so next on to the data analytics part. This is much different than the previous part of the presentation. But basically, we have a very large data set with a lot of parameters measured per sample. And there's a lot of interest in using in developing models to predict some of these indicators that are more costly and time consuming to measure. So most of this is going to be about using random forest to model available water capacity and base protein. But before we do that, just a little bit of information about this very massive national Cornell soil health database. This is the sample sizes that it looked like a couple years ago. The subset has some GPS information, and I'm sure some of you remember Marcio Nunes. We're still working very closely with him. Now he's with the USDA ARS and other USDA ARS and NRCS researchers to help use this information to advance soil health testing and interpretation across the country. So without digressing too much from the random forest, I just want to show you this. This was just, the final version was just accepted yesterday. And so we have moving forward with the National Comprehensive Soil Health Assessment, and SMAF was kind of the USDA ARS's approach and CASH approach. And now we have this acronym SHAPE, Soil Health Protocol, Assessment Protocol and Evaluation Tool, which kind of merges those with some more advanced statistical methods and a much larger database that's nationwide. So this data set had something like 27,000 observations, combining our observations, published studies, and a lot of NRCS data. So basically what this is here, I'll just read this sentence verbatim because it says it more concise than I could say it. So the scoring, basically using some Bayesian model estimates to define this conditional cumulative cumulative distribution function for each cohort of samples. So we're using mean annual temperature data, mean annual precipitation data, suborder, and soil texture. And right here, I've just kind of honed in on, there's basically 5 of these figures in the table for each different soil texture group that they have. And the suborder S4 is kind of like what we have in New York State. But you can see the influence of the different climates. So if you're in a very hot and dry climate, 2% soil organic carbon is extremely good. If you're in a maybe climate like ours, then it might be around 50%. And then if you're in a much colder climate, then that might not be as good. So this is a really nice approach that allows the same scoring approach to be able to be used for any point in the continental US. Okay, so now to the actual prediction of available water capacity. Regression is a wonderful tool, but if you're really into getting as good prediction as you can, traditional linear regression techniques can only get you so far with prediction. You can see us maybe explaining 61% of the variance. Seems like sand is the most important. We have a really big database here. So we've been using random forest to predict available water capacity for 2 years in the lab. We're explaining about 10% more variation. And the RMSE kind of tells us that that the prediction is about as good as the accepted variance that we would accept between replicates in the lab. So it's a pretty good model. And we have a lot of the predictors from the soil health assessment in there. So let's, if you haven't heard of random forest, it's becoming a very popular machine learning nonlinear method. And basically these decision trees, I made a decision tree for AWC. is kind of the basic building block of random forest. But this isn't actually the model we use, because the model is more complicated than that. Random forest is basically taking a random subset of the predictor variables and a random subset of the data and making a lot of these trees, about 500 trees in my case. And then the random forest assembles the many trees to come up with the best prediction. And this is, if you're into random forest, this package, Random Forest Explainer, is really wonderful, because unlike linear regression, which is much more mechanistic, you can see the coefficients for the model for each variable. This is a little bit more black box, so there's some really nice diagnostics you can get, and which makes sense with how much we know texture controls AWC. We see sand and silt are really high up there. This is basically that the when it's running, the summarization of running those 500 iterations of the random forest. So if we're at node 0, it means that the sand and the silt tend to be the best indicators for that top of the decision tree, and followed by organic matter and clay. And another tool that I use to help visualize the effect of these different predictors in the model is to kind of use a made-up data set where just one thing changes it at once. Then you can run the model and see basically how that single indicator's being used in the model. So this helps us visualize it. And this is nice from soil physics, and Harold and Bob could explain this better than I can. But basically, once you get, well, basically all those coarse textured soil, those soil texture classes, those coarse soil texture classes, a lot of them, that definition is at 50%. So once you get more than 50% sand in the soil, that's when you're starting to get sand against sand and creating more of those bigger pores, which have a negative relationship with available water capacity. Also positive, no. And also of huge interest is, we talk about this a lot in extension, and people want to be managing their soils for greater available water capacity. But at least within our model and some other work has shown, this work helps us visualize that soil organic matter has a much greater effect of improving available water capacity in coarse textured soils compared to silt loam, which already has a pretty good amount of available water capacity. And I'm going to stop there. It seems like that's the perfect time to cut it off. I had ACE protein, but we'll just skip that. So plenty of time to take questions if people have questions. And definitely want to acknowledge our large team: Harold Van Ness, Bob, Kirsten Kurtz, David Wolf, Galia Barshad, who made some of those beautiful maps, Marcio Nunez, who I'm still closely collaborating with, Katie Sommerfeld, Dennis Kirby in Orleans, Debbie Aller in Long Island, Franklin Egan from the PASA Inspiration, and all the wonderful people in the lab.
Thanks a lot, Joseph. That was excellent. The floor is open. I think, David Rossiter, you have your hand up, so why don't you go ahead?
Hey, Joseph, thanks a lot. Very interesting. Your very last thing you presented, it seems you could convert that to actually a value of this of drought resistance. If you fed this into a simulation model and you say, well, with certain climate scenarios, if you had this better available water capacity, you're actually going to get this much higher yield. So you could go one step further with the improved water capacity. What does it really give me? So you've shown the water capacity. What is really the effect of this? And the bigger effect presumably would be getting through those drought periods in August, for example. So it's just an idea, because I think one thing missing in this whole soil health story in general is the link to really the the final value of having a healthier soil. And this would be one way to actually get to that. So it was just a comment. Now that's enough for, you don't really have to respond yet.
No, that's great. No, that's definitely one of the challenges of just requires so much information and such large data sets to be able to pull together information. And I think hopefully with more of these regional partnerships, we can start hopefully maybe getting some of those linkages and linking this information to yield, which is, it's really important.
Johannes, you have your hand up.
Thank you. Thanks, Joseph. Good to see all this to come together. It's so exciting. Great presentation. My question follows up on what David just said. The quantification of what's good, what's bad, and how does that translate into a management in any quantitative way has been a little bit of a stumbling block. So what do you think, or from my vantage points, what's the response from farmers in New York or elsewhere, but maybe specifically in New York State, that see these values and in the first instance, as you mentioned, compare them to their neighbors or the cloud of their neighbors. But how do they translate that into action? And what can you learn from these responses of farmers that maybe translate at some point in a quantitative recommendation system that says, well, if you have that value in active carbon, you have to change your management to X?
Yeah, it's definitely a complicated question. And I think I really like the PASA framework where in their report they have those steps that you would take to change your management. Oh, my days of living cover is much less than my neighbor's days of living cover. So I think those tools can also be helpful in making those linkages more explicit. But I think we're just at the beginning of maybe providing more useful information for peers, for people to help pique people's interest more. Because if it's just a score based on soil texture, that's maybe not as interesting as having having data from your peers. So I think, yeah, I think the jury's still out, and we're working to try to make it useful and useful to everyone, but it's not easy.
Thanks, Joseph.
Thanks, Johannes. There's a question in chat from Peter Woodbury. Thanks, Joseph. Can you say anything about bulk density and also address the issue of depth of sampling for these samples?
Yeah, so can't say anything really about bulk density. It's possible someone could trample the soil and then their 6 inches is maybe different than really 6 inches, but a lot of the samples collected in this are collected by professionals that know what they're doing, and the sampling procedure is pretty clear. All the samples are 0 to 6 composite samples, but sampling's really important if you wanna. We talk about sampling a lot because unlike nutrient analysis, where it's much cheaper and you can do grid sampling across a 100-acre field. For this more detailed analysis, you really want to be targeted because the variability in the field can swamp the pattern. Yeah, so I can't say anything about bulk density, but depth of sampling should be 0 to 6 inches.
Thanks, Joseph. We have another hand up. David, go ahead.
Hi, Joseph. Thank you for a nice presentation. I was just wondering, is there any interest on your part or vice versa with the forestry industry and adding some depth to site indices that are traditionally used as a gradation of the quality of or the cutting cycle for cohorts or different cuts in logging?
I don't personally want to go there because it's a whole different there's people that have done a lot of work in forest soil health. Yeah, it's just, for me, it seems like such a dramatically different system that at least I want to try to focus on more agronomic, and maybe perennial fruit is the furthest. I'm currently interested in going just because the considerations are so different, and I've seen some very different relationships with forest soil health and maybe more agronomic soil health, with bulk density not having effect on tree. I don't know. It's just it's more complicated, or it's much different than the agronomics. But
Thanks.
there is some good work done out there on that.
We've got a couple, well, Harold, I guess, Harold's hand is up, and Joseph will release you in a second. I know that we're now over time, but Harold, please go ahead.
No, I just wanted to make a quick comment. I can't help but reading the subtitles and the speech recognition. It recognized mycorrhizal fungi. So you're pronouncing it correctly, Joseph, and I'm impressed with the speech recognition software. We've made progress, folks.
But I'm not from Africa, I'm from Ithaca. Joseph, one last question from my side, a short one, and then we'll close. But I'm curious if you have crop performance data from these sites, and if you put that into an integrative analytical framework.
We don't have crop yield from those sites. I think that would be, I mean, hopefully it would be maybe a next level where we can collaborate more closely with Orleans soil water conservation districts and propose that. But a lot of the information's out of our hands, so it requires coordinating and making these partnerships to link the data with yield information or plant performance information. So that's definitely, I think, yeah, is critical but difficult.
Great. Joseph, thank you so much. That was a really tremendous presentation. So on behalf of everyone, thanks everyone for coming today. The attendance was great. Very well done, Joseph. And thanks everyone for attending.
Awesome. Thank you so much, everyone. Have a great rest of your week.
This has been a production of Cornell University, on the web at cornell.edu.